Swayam Prabha. Digital India. Educated India.
In this section, we are going to discuss a few more advanced techniques which are widely utilized in the field of biotechnology in different areas. These will include polymerase chain reaction, we call it PCR, automated DNA sequencing methods, and ELISA, that is enzyme linked immunosorbent assay. We will begin our discussion with polymerase chain reaction. Now, this technique was developed in 1983 by Carey Mullis, for which he received the Nobel Prize in chemistry in 93. Now, PCR is a technique that takes a specific sequence of DNA of small amounts and amplifies it to be used for further testing. Now, basic technique here in very simple case is PCR amplification requires a small amount of nucleotide sequence at each end of the region to be amplified. So, whichever sequence you are going to amplify, you require knowledge of nucleotide sequence at each end of this region. Now, oligonucleotides complementary to that sequences are synthesized, and it is typically around 20 or so nucleotide long. These oligonucleotides are used as primers for enzymatic amplification. Now, here a reaction mixture is set up containing a sample of DNA that includes the region to be amplified, the primers in large molar excess, deoxynucleoside triphosphate, or we call it DNTPs, and a heat stable DNA polymerase for the enzymatic synthesis. So, the mixture is heated to a temperature sufficient to melt or separate the strand of the sample DNA. Remember that in cells this function is performed by an enzyme, that is helicase, but here outside the two DNA strands needs to be separated, and that is called DNA melting. At particular temperature they will separate from each other. Now, after they are separated, they are cooled to a temperature which is low enough for the primers to anneal to the DNA, that is at the two ends where in a particular region which needs to be amplified. Then the mix is incubated at a temperature sufficient for the polymerase to synthesize a complementary strand to each piece of sample DNA. Now, starting with the primers and using the DNTPs provided. So, the number of copies of the target region is doubled. After this round of DNA synthesis, the mixture is heated for strand separation again, cooled again for primer annealing, and again incubated for another round of DNA synthesis, and likewise many cycles will be performed. So, each time the cycle is carried out, there is a doubling of the number of copies of the target region between the primer annealing sites. After say 25 or 30 cycles, there is theoretically amplification of over 10 raised to power 9 fold after 30 cycles. Now, applied even to a single DNA molecule of 1 kilobase pair, that would generate 1 nanogram of DNA. So, it is about sufficient to see in a gel or for any other application thereafter. So, in very simple terms, this figure explains the technique here, the simple technique. So, in starting what you have is, you have a piece of DNA, that is double stranded DNA, and there are primers, DNTPs and polymerase which are mixed with in a tube. Now, the first step is the DNA melting or denaturation step. So, the two DNA strands are separated here. Once they are separated, then temperature is lowered to say around 60 degrees Celsius. Now, this temperature will depend upon the melting temperature of the primers. So, the primer will anneal at this temperature, which is little lower than the melting temperature of primer, and they will anneal at the right complementary place. So, DNA synthesis, then temperature again heated to 72 degrees Celsius, and the DNA synthesis takes place. Now, at 72 degree around, DNA polymerase will synthesize the complementary strands. Then again the cycle will be repeated many times, and many double stranded DNA amplified fragments will be obtained. Through these three steps, the DNA amplification or polymerase chain reaction will take place. All right. So, let us little bit get into details of the PCR technique. When amplifying a small segment of a large double stranded DNA template, the desired fragments first appear in the third cycle of PCR, as we have seen in the figure. The amplified products then accumulate exponentially as described in this equation, where you can see NF equals N0 1 plus y raised to power n, where NF is the copy number of the amplified sequence after n cycles of amplification, and N0 is the initial copy number of the target sequence in the DNA template. Y is the efficiency of the amplification per cycle. Now, what are the essential components of polymerase chain reaction? As we have seen, one is thermostable DNA polymerase to catalyze the template dependent synthesis of DNA. Then a pair of synthetic oligonucleotides, that is primers. Then careful design of primers is required to obtain the desired products in high yield and to suppress amplification of unwanted sequences and to facilitate subsequent manipulation of the amplified product. Then DNTPs which are required, and standard PCR contains equimolar amounts of the 4 DNTPs, DATP, that is DTTP, DCTP and DGTP. Then there is a requirement of divalent cations, because all thermostable DNA polymerases require free divalent cations, usually magnesium, for activity. They are kept in a buffer to maintain the pH, like TrisCl adjusted to pH between 8.3 and 8.8 at room temperature is included in standard PCRs at a concentration of around 10 millimolar. Monovalent cations are also included in standard PCR buffer like KCl, which is like works well for amplification of segments of DNA greater than 500 base pairs in length. Raising the concentration to 70 to 100 millimolar improves the yield of shorter DNA fragments. Then you require template DNA. The template DNA containing the target sequence can be added in single or double stranded form. Closed circular DNA templates are amplified slightly less efficiently than linear DNAs. Now, let us discuss about thermostable DNA polymerase. The most common enzyme which is utilized in PCR is Taq polymerase. It is a DNA polymerase isolated from thermophilic bacterium Thermus aquaticus, and this enzyme, which the bacterium uses for cellular DNA synthesis, has a temperature optimum of at least up to 80 degrees Celsius and is not readily denatured by the repeating heating and cooling cycles that are needed in the amplification process. It is generally produced by expression of the gene in E. coli, and enzyme has a 5 prime to 3 prime DNA polymerase and 5 prime to 3 prime exonuclease activities. So, it will polymerize about 50 to 60 nucleotides per second. However, the enzyme has a number of properties that may be disadvantageous, like Taq polymerase has no proofreading, that is 3 prime to 5 prime exonuclease activity. If a mutation happens in an early cycle, then a large fraction of PCR products will have the altered sequence. Then Taq polymerase has relatively low processability, which places a maximum size limit of molecules that can be amplified using this enzyme, which is typically 2 to 4 kilobase pair or maybe little lower actually. Now, Taq polymerase is not fully heat stable. It has the half life of about 40 minutes at 95 degrees Celsius, which means that there will be significant loss of activity over the 30 cycles used in a typical PCR experiment. Then Taq polymerase incorporates an extra A residue and this is incorporated on the 3 prime end of the molecule synthesized and is not template encoded. So, this overhang residue may help in cloning the products of PCR, but it depends on what strategy one is utilizing. Now, apart from Taq polymerase, there are number of other polymerases which are available from other thermus species like TF1 and TTH enzymes from Thermus flavus and Thermophilus respectively are available, which have 3 prime to 5 prime proofreading activity also. Polymerases are also available from other bacteriums like Archaebacterium also, and many of these enzymes have proofreading activity. These include like TLI from Thermococcus litoralis, PFU from Pyrococcus furiosus and deep vent from Pyrococcus species. These are enzymes which are available and these are sometimes more stable than the Taq enzyme. So, when you are performing, one is performing polymerase chain reaction, it is performed on a thermocycler and programming can be done like PCR. It is a iterative process which involves 3 steps, and these 3 steps can be programmed so that machine can run for particular number of cycles. Now, these 3 steps, one like we have discussed, first is denaturation, where double stranded DNA template denatures at temperatures of around 95 degrees Celsius, which is the highest temperature that Taq polymerase can really endure for around 30 or odd cycles. So, in the first cycle of PCR, denaturation is carried out for 5 minutes to increase the probability that long molecules of template DNA are fully denatured. The second step is annealing of primers. So, what is done for annealing of primers, temperature is brought down and this is a very critical step if the annealing temperature is too high, then oligonucleotide primer anneal poorly and yield of amplified DNA is low. If the annealing temperature is too low, non-specific annealing of primers may occur, resulting in the amplification of unwanted segment of DNA. So, annealing is usually carried out at 3 to 5 degrees Celsius below the calculated melting temperature of the oligonucleotides, that is primers. So, after annealing, that is when primers are annealed at a particular specific sites. Third step is extension, that is extension of oligonucleotide primers. Now, this is carried out at the optimal temperature for DNA synthesis catalyzed by the particular thermostable polymerase. Like in case of Taq polymerase, it is around 72 degrees Celsius. Now, in the first 2 cycles, extension from one primer proceeds beyond the sequence complementary to the binding site of other primer. Then in the next cycle, the first molecule are produced whose length is equal to the segment of DNA delimited by the binding sites of the primer. So, from third cycle onwards, the particular segment of DNA amplifies geometrically, whereas longer amplification products accumulate arithmetically. So, extension is carried out for 1 minute for every 1000 base pair of product. That is a kind of a particular efficiency. For the last cycle of PCR, extension is carried out 3 times longer than the previous cycles to allow completion of all amplified products. So, the last extension step is little longer. Now, number of cycles required for amplification depends on number of many factors, like say number of copies of template DNA present at the beginning of the reaction, that is important. Then efficiency of primer extension and amplification. So, once established in the geometric phase, the reaction proceeds until one of the components becomes limiting. So, at this point the yield of specific amplification product should be maximal, whereas non-specific amplification product should be barely detectable. Now, this is the case after 30 cycles of PCR containing almost like 10 raised to power 5 copies of target DNA. At least 25 cycles are required to achieve acceptable levels of amplification of single copy target sequences in mammalian DNA templates. A typical set of reactions might have like in initial melting carried out for 5 minutes at say 94 degrees Celsius, then 30 cycles each comprising like it is a repetitive like melting for 1 minute at 94 degrees Celsius, renaturation for 1 minute at 60 degrees Celsius, and then DNA synthesis for say 1.5 minute at 72 degrees Celsius, and finally, final extended round of DNA synthesis for 5 to 10 minutes. The primer design is an important part of PCR experiment, and there are several computer programs available now to suggest suitable primers. And general guidelines is 1 length. A short primer may offer sufficient specificity while amplifying using a simple template such as a small plasmid. A long primer may be required when using say eukaryotic genomic DNA as template. So, in practice like we said around 20 to 30 nucleotides are satisfactory. Now, members of a primer pair should not differ in length too much, like greater than 3 bases, and base composition also has to be looked in. Like if you have higher G plus C content, that will be a problem. So, it should be around 40 to 60% with an even distribution of all 4 bases along the length of the primer. If there is very low GC content, then also there is a problem because the melting temperatures will be lower actually. Then mismatches where primers do not need to match the template completely, although the 3' end of the primer should be correctly base paired to the template rather than 5'. It is often beneficial to have C or G as the 3' terminal nucleotide. This makes the binding of the 3' end of the primer to the template more effective. Some mismatches in the body of the primer sequences may be allowed, particularly during mutation, like when somebody is trying to mutate a single base or try to introduce say restriction sites. So, that way sequences can be incorporated at these ends that contain restriction endonuclease recognition sites to facilitate subsequent cloning and manipulation of the PCR product. Melting temperature is the temperature at which the 2 primers can associate with the template and should be relatively similar, so that they both can bind at the same time, as temperatures are being lower during annealing. If the temperatures of both primers are not same, then they will not bind at the same time and there will be problem in extension of both the strands. Now, internal secondary structure formation should be avoided. Primer sometimes may fold back on itself and not be available to bind to the template, and then proper precautions or has to be taken to avoid this. There could be cases where primer annealing could take place and to avoid the primers being able to anneal to each other, extension by DNA polymerase of two self annealed primer leads to the formation of a primer dimer, and this will be very very efficient templates for amplification in subsequent rounds of PCR, as they are like small actually. So, primer annealing has to be avoided. Now, this was the basic technique of PCR, polymerase chain reaction, with three main steps here, and many like enzymes, particularly it started with Taq polymerase, but other enzymes could be also utilized. And PCR has a number of applications in various areas of biotechnology and other areas as well. Now, why it has so many applications? One is it is a very convenient technique. Amplifying a DNA segment was very difficult before this thermocycler technique came in, or PCR technique came in. So, PCR is automated. A typical set of cycles can be carried out on many separate samples simultaneously in a few hours, and starting amounts of DNA can be very small. Like in forensic science, it is like when you get a blood sample or hair sample, from that also amplification can occur. So, there are so many applications of PCR technique or this particular method, and we are just discussing some of them here. One is DNA sequencing. Now, DNA this PCR in the presence of dideoxynucleoside triphosphate or DDNTPs, we will be discussing about that in the next section. That is a chain termination reaction or method, where DDNTPs are utilized for chain termination for sequencing in Sanger's method. So, this allows DNA sequencing reactions to run successfully with very small amounts of template. This is known as cycle sequencing. It requires a specially developed enzyme that combines the properties needed for sequencing with thermostability. So, one application is DNA sequencing, and this could be done by different methods, including the Sanger's method. Then PCR technique is widely utilized in diagnostic. It is a diagnostic tool actually. Like for example, in the identification of specific genetic traits or for the detection of pathogens or food contaminants, it could be utilized. One of the first applications of PCR was to genetical diagnosis for sickle cell anemia actually. The sickle cell anemia in the beta globin gene destroys the restriction site, and the test involved PCR amplification of this region of the genome and analysis of the PCR product for the presence or absence of this restriction site. PCR products can be analyzed by use of restriction enzymes, although it is rare for a mutation to create or destroy a restriction site, but this could be done. Determining whether an oligonucleotide probe specific for a particular allele is able to hybridize to PCR products. Then electrophoresis could be utilized, like screening for mobility changes caused by sequence differences between a PCR product carrying a mutant allele and wild type molecule can be done. Then also sequencing can be done, like PCR products can be directly sequenced by DNA sequencing methods. So, the test for the presence of a particular pathogen here, like in diagnostics, can be made using primers specific for the genomes of those organisms, and this permits detection at extremely low levels, like infections could be detected at a very initial stages rather than advanced stages. A similar approach can be used to detect DNA from contaminating sources in food. So, the PCR is also widely utilized in forensic science. The ability to amplify DNA from regions of the genome that are highly polymorphic and therefore, which are variable between the individuals. This could be starting with samples containing a very small amount of DNA like single hair or traces of body fluids like say blood or semen. This could lead to the applications in forensic work. A number of polymorphic regions have been utilized as targets for amplification, including like D loop of mitochondrial DNA, which is variable between individuals, then tandemly repeated mini satellites, also known as variable number of tandem repeats, VNTRs, then microsatellites used in conventional genetic fingerprinting, human leukocyte antigen sequences, they could be utilized for these applications. Now, PCR can also be utilized in present day population genetics. The ability to amplify material rapidly from a large number of DNA preparations leads to the application in population genetics, allowing for example, the determining of frequencies of a particular allele in a large collection of individuals. There are number of PCR techniques that will provide information from many parts of the genome simultaneously. Now, one such is random amplification of polymorphic DNA, or we call it RAPD. So, this analysis utilizes relatively short primers that will anneal to many different sites in the genome under study, producing many different bands when the PCR products are analyzed on electrophoresis. Now, the similarities and differences between the band patterns generated from genomic DNA of different organisms provide information on their genetic relatedness. Another technique is there which involves amplification of large set of restriction fragments of genomic DNA, whose sizes can be compared on electrophoresis, and this is termed as amplified fragment length polymorphism or AFLP. So, PCR is also used in population genetic studies like we said of microsatellites or other like tandemly repeated sequences, single nucleotide polymorphism and transposons could be studied. A particular advantage of using PCR in population genetic studies is that with appropriately designed specific primers, it may be possible to amplify DNA from one organism that cannot be separated from others. So, such as particular bacterial strain in a mixed population that is very difficult to separate, but through PCR it could be done. So, this approach therefore can be used to study genetics of bacteria or other organisms that cannot be cultured axonically. Now, another application of PCR is in archaeology and evolution. PCR can be used with old material as well as more recent samples, and it is often possible to amplify ancient DNA from museum specimens and archaeological remains. Multiple copy sequences such as mitochondrial DNA or chloroplast DNA are a particularly useful target, and nuclear DNA could also be retrievable in certain cases. So, comparison of polymorphic sequences from the ancient DNA with the sequences observed today allows inferences to be made about the origins of a particular population of species. So, these were some of the applications which makes PCR a widely used technique. Now, there are certain modifications in the technique for improving specificity. One of the most frequently encountered problem is the annealing of the primers at the wrong location or to generate the wrong, and this will generate wrong PCR product. So, it may be possible to reduce this problem by raising the annealing temperature in the cycle conditions or altering the ionic concentration in the reaction mixture, but there are number of other approaches also which we are going to discuss. One approach could be which is called hot start PCR. So, as soon as the PCR reagents have all been mixed up together, it is possible for the DNA polymerase to start synthesis, although the optimum temperature is very high, and this may happen while the reaction mixture is being heated for the first time and is at a temperature which is low enough to allow non-specific annealing of primer to template, generating a range of non-specific products. So, this problem would be could be prevented if DNA synthesis takes place or does not take place until the first cycle had reached its maximum temperature. So, this is the basis of hot start PCR, that to prevent the DNA synthesis to occur until it reaches the highest or maximum temperature. So, in the simplest form, the DNA polymerase is not added to the reaction tubes until they have reached the DNA melting temperature of the first cycle, but this is like not possible, though you can do it, but it is like then you have to mix again and again. A more convenient approach would be to have one of the reaction components in a form that is unavailable and until that particular appropriate temperature has been reached actually, and this can be achieved by incorporating the polymerase or say the magnesium salt in wax or some capsule like structure, and which these beads or this particular material melts at the appropriate temperature, allowing the reaction to start. A different approach could be to have the polymerase inactivated at the start by complexing it with an antibody, and then antibody denatures at high temperature, so that polymerase could be functional. Another approach could be touchdown PCR. Now, touchdown PCR, here the annealing temperatures used in conventional PCR is usually several degrees below the maximum at which primers can remain bound to template to ensure stable binding. However, this use of lower temperature permits a small amount of mismatching between primers and template, which may allow primers to bind to incorrect sites and generate the non-specific or spurious products. Now, the effects of this can be reduced with touchdown PCR. What is done here? In this, a high annealing temperature is used initially, at which even correct binding may not be possible. The annealing temperature is reduced in subsequent rounds actually. So, there will come a certain point at which correctly matched primer template annealing will be possible, but incorrect matching is not. So, DNA synthesis can therefore start. Now, although later cycles may be under less stringent conditions, the earlier cycles will have been carried out under the most stringent conditions, and the desired products will be the most abundant. Another approach could be nested PCR. Now, here two successive PCRs are carried out. Now, in first PCR, it utilizes a conventional template. The product of the first PCR are then used as the template for the second PCR with primers that are designed to anneal within the desired product of the first PCR. Now, although the first PCR may generate some non-specific products in addition to the desired product, it is unlikely that the non-specific product will also contain annealing sites for both the primers used in the second PCR. Thus, only the desired product from the first PCR are likely to be suitable templates for the second. So, here it is shown here in this figure, you have a DNA template and you will amplify with first set of primers here and amplification will occur. Now, what is done? The second pair of primers are annealing between the product of the first PCR, and then it is re-amplified. So, that only those which sites, because sites is within the first product. So, that will be very specific. Now, there is another term called making hard copies actually. So, having amplified a specific piece of DNA, it may be convenient to insert a copy of the products into a vector for subsequent cloning and maintenance by conventional means. This is sometimes called making a hard copy actually. So, the PCR product can be treated as a blunt ended DNA molecules for cloning purpose actually. Another approach is to digest the PCR products with restriction enzymes prior to cloning. Now, if suitable restriction sites are not present within the DNA sequences to be amplified, then it is possible to incorporate. So, either it could be done by blunt end or there could be other like restriction sites which can provide for the cloning. So, then if they are incorporated, this can be done by inclusion of an appropriate sequence containing a restriction enzyme recognition site at the ends of the primers, and when they are synthesized, they will the new DNA will have those restriction sites at the ends. So, the primers anneal to the target DNA with the restriction site sequences unbound, and the intermediate molecules that are formed will have the restriction sites present at one end, and all the target length DNA molecules will have to have the sites at both ends. So, the amplified molecules can then be cut at their ends with the appropriate restriction enzymes, and then fragments can be cloned easily. So, here like it is shown here, if you can see these are not really pairing here, but these contains the restriction sites, and finally, what the product will have the two restriction sites here due to the primers containing the restriction sites actually. So, the target length molecule with restriction site at both ends is obtained and which can easily be cloned. A third approach to making hard copies of PCR products relies on the fact that polymerases such as the Taq enzyme that lack proofreading activity, and they add an extra unpaired A residue or at the 3 prime end of the molecule they have synthesized. So, a PCR product can made using such an enzyme will therefore have unpaired overhanging A residue. So, at each end, and this allows the molecule to be ligated to a linearized plasmid molecule that has unpaired T residues. Remember, A will pair with T and C will pair with G. So, so this could be another approach where Taq polymerase if you are utilizing and there are A overhangs, then the plasmid can contain T overhangs and they could be cloned in there. Another approach is to incorporate topoisomerase 1 recognition site into the PCR primer, and vectors with covalently attached topoisomerase is then added, and the enzyme cuts the PCR product at the recognition sites and joins it to the vector. Then there is another term called inverse PCR. Result of the standard approach outlined so far as we have discussed is the amplification of sequence between the primer annealing sites, that is you have two primers at the ends and in between that the particular segment of DNA is amplified. However, it is also possible to arrange for the amplification of sequences outside the primers, and this technique is called inverse PCR. Now, the sample DNA is first cut with an enzyme outside the region whose sequence is already known. Now, the resulting linear molecule are then circularized by ligation under conditions that favor intramolecular reactions rather than intermolecular reactions. A second restriction digestion is then done using an enzyme cutting within the region of the known sequence. The result is now that the first fragment containing this sequence has been turned actually inside out, leaving known sequence on the outside and the material that had previously been flanking it within. So, primers complementary to the known sequences on the outside of the molecule now be used to amplify the region of interest between them. This technique is especially powerful when combined with transposon tagging actually. If a previously characterized transposon has inserted into a gene of interest, the gene can be amplified using the inverse PCR, and primers from transposon sequences could be utilized. Now, if you see here this figure on your screen explains this. So, there is a region of known sequence here in the middle, and when you cut the DNA, this region is in here and this is ligated, self ligated, and what you get is that known region is in here. Now, what is done is it is digested with enzyme which is cutting within the known sequence. So, what you have done is first you have circularized it, and then there is a known region is in this particular place which was at the center earlier, and it is being cut within the known sequence. When you cut it within the known sequence and linearizes it, this known sequence which was at the center is now at the ends, and then primers against this could be utilized. Now, since you have known DNA ends, and this could be utilized for PCR amplification. This was about inverse PCR. Now, there are other types of PCR like in situ PCR. Now, it is possible to carry out PCR using permeabilized tissues actually, say thin sections on a microscopic slide. So, what it requires is a specially adapted PCR machine to accommodate these slides actually. If the PCR product can be detected, then this allows one to identify where in the tissue the target nucleic acid is located. Although this can be used to identify the location of particular DNA species in tissues, it is very often combined with reverse transcriptase to identify the location of particular transcript actually. Quantitative PCR could also be done, and it is possible to use PCR to estimate the abundance of a particular nucleic acid molecule in a sample. Say if we are interested in an RNA molecule, it will be necessary to start with the reverse transcriptase PCR or RT-PCR to make a cDNA copy. Now, there are two main approaches here for estimating the abundance of a molecule. One is to use the standard PCR and visualize the amount of the product of interest by electrophoresis technique, like comparing it with suitable controls or standards. So, this is like in at the end of the experiment you are measuring the amount of target sequence. Now, second approach could be to quantify the reaction in real time, that is while the PCR is progress. So, the real time PCR is a very important technique in terms of giving an information about like in terms of quantitative PCR, and this can be done in two ways. One is fluorescent double stranded DNA binding dye like say cybergreen is present in the PCR, and as the double stranded DNA product accumulates, the amount of fluorescence from the dye increases, and this can be detected. Now, the experiment requires a PCR machine that is also equipped with a fluorescence measurement facility. So, this approach is adequate if the PCR generates the products of interest very specifically. Now, because the method simply detects double stranded DNA, it measures the amount of PCR product at a given time regardless of whether it is from the correct region or not actually. So, here like through fluorescent probes like double stranded DNA binding dye could be utilized as one approach. In the second approach to real time PCR, it allows detection of a specific product rather than double stranded DNA in general here. And what it does is it utilizes a specially synthesized probe oligonucleotide, and this probe is designed to anneal within the region to be amplified and carries a fluorescent reporter dye at one end and a quencher at the other end of the molecule. So, what happens is if the quencher and the reporter are in close proximity, that is attached to the same oligonucleotide, then the quencher stops the reporter from fluorescing. Now, during PCR, the probe will anneal two single stranded DNA within the target region, and when the polymerase meets the annealed probe, the 5 prime to 3 prime exonuclease activity of the enzyme degrades the probe, liberating the reporter from the quencher, and thus the fluorescent reporter accumulates during the course of PCR. So, this could be then detected. So, here is very simple depiction of that. What you have is, if you can see on your screen, there is a reporter adjacent to quencher. So, when this reporter this quencher is there, the no fluorescence will be obtained or seen, but as soon as DNA polymerase comes and it kind of removes this, then this is freed here and polymerase will separate the reporter from quencher and the fluorescence could be detected actually. So, this is the approach here for real time PCR. Now, an alternative method for specific products detection in real time PCR uses two oligonucleotide probes that anneal to adjacent sites in the target DNA. One oligonucleotide is tagged with a molecule that absorbs light and the second with a molecule that is able to accept energy from the first and re-emit energy at a different wavelength. Now, if the two oligonucleotides can both anneal to the target DNA, then the energy absorbing and energy emitting molecules are brought into close proximity and energy transfer is possible. Now, if the oligonucleotides are not brought together in this way, then the little energy transfer can take place. Thus, measurement of the amount of fluorescence from the second tag provides a measure of the amount of the target DNA. So, here it is like as you see these two targets when they come together, that is they are brought into the proximity, then only the particular fluorescence measurement of the amount of fluorescence is provided actually. Mutagenesis could also be in PCR reactions, mutagenesis can also be performed because perfect matching between the primer and the target DNA is not necessary. So, this could also be one of the methods to make sequences with certain mutations. Now, provided that this allowance is made for this in determining the condition for primer annealing. So, primer annealing should not be affected if you are putting in a primer with mismatches. PCR can therefore be used to engineer specific mutations in the amplified sequences by constructing a primer that corresponds to the mutated sequences rather than the original. There are other forms of PCR like asymmetric PCR. By reducing the amount of one of the two primers, it is possible to arrange for preferential application of the one of the two strands, resulting in a preparation of single stranded DNA, which has a number of uses in molecular biology. So, asymmetric PCR can be another form where a single stranded DNA could be there. Then anchored PCR. Anchored PCR is applied when only one piece of sequence for the region of interest is known. The aim is to attach the region to be amplified to a piece of known sequence and then to use that as the second priming site. And there are two types, two ways in which this can be most easily done. One is to fragment the sample DNA and ligate it to molecules of the known sequences such as vector, and this known sequence is used as the basis for designing one of the two main PCRs. And second could be to add tails enzymatically to the sample DNA or the molecules produced after the first round of synthesis. For example, a run of say G residues can be added by treatment with terminal deoxynucleotidyl transferases or some other can be used, and this could be combined with RT-PCR. This forms the basis of rapid amplification of cDNA ends. So, what is done is say through an enzyme you have put in G and 2' deoxyguanosine 5' triphosphate or oligo DC can then be used as a primer, because it will anneal to the oligo DC. Combined with RT-PCR, this forms basis of rapid amplification of cDNA ends, or it is called race. So, this picture kind of explains that there is a single sample DNA cut and ligated to anchor, and then PCR with primers for sample DNA and ligated anchor is performed, and this way you can get the full sequences actually. So, this is called anchored PCR. There are other forms like emulsion PCR can be another form. In this technique all the reagents are incorporated inside the lipid droplets and PCR is carried out on a much smaller scale. Advantages could be to it is possible to increase and decrease the temperature of small droplets very quickly. If each droplet contains a single template molecule at the start, then all the products in an individual droplet results from the amplification of single template molecule. This is also called droplet PCR. Then there could be another form could be isothermal amplification. The repeated heating and cooling required by PCR limits how quickly the process can be carried out. In addition to this, the cost of PCR machine is not low. So, loop mediated isothermal amplification has been developed, which allows templates to be assembled amplified at a constant temperature, say around 65 degrees Celsius. It uses a DNA polymerase with strand displacing activity and avoids the need for heating to high temperature. There is a particular interest in the use of this method for the detection of pathogens outside of specialist laboratories, as it is a rapid and avoids the need for expensive PCR machines. So, these were some of the different modified techniques and different approaches which have been developed from the simple basic techniques of PCR. So, in this lecture we have discussed about polymerase chain reaction, basic technique and modified versions of this technique. Also we have discussed about the applications of various applications of PCR technique. In the next lecture we are going to discuss about two more techniques here, that is automated DNA sequencing methods and ELISA. Thank you.